Hello friends, today we are going to discuss about one of the important topic in developmental biology, about spermatogenesis. About spermatogenesis. You know, the most or fundamental or essential steps in sexual reproduction are number 1, gametogenesis, number 2, fertilization, number 3, development of zygote. Here gametogenesis means spermatogenesis and oogenesis. Here gametogenesis means the development of sex cells or gametes from the germ cells is gametogenesis. Here gametogenesis is germ cells are spermatogonia in case of male, whereas oogonia in case of females, and the spermatogonia becomes spermatozoa, whereas oogonia becomes egg or ovum. Most of the part, the fundamental principles operating in both spermatogenesis and oogenesis are almost same, and the 4 essential steps in gametogenesis are: number 1, extragonadal origin of primordial germ cells. Number 2, proliferation of germ cells by mitosis. Number 3, meiosis. Number 4, structural, simply called maturation. Number 1, extragonadal origin of primordial germ cells. Then number 2, proliferation of germ cells by mitosis. Number 3, meiosis. Number 4, maturation of, that means sperm or ova. These are the 4 basic principles in both spermatogenesis and oogenesis, what we call fundamental operating principles in gametogenesis. Here primordial germ cells is very important. Extragonadal origin. Generally we believe that, say, spermatogonia is a mother of sperm cells, oogonia is a mother of ova or egg. We believe that these two, I mean, both are originated in that means their precursors originated in their respective gonads, say testes and ovaries, but it's not like that. Extragonadal, that means the primordial germ cell, which is called earlier precursors of both sperm and ova, located, that means originated outside their gonads, that means testes and ovaries, what we call extragonadal origin of primordial germ cells, and they form early in the embryonic development, that means during early fetal development. After they form, they proliferate and migrate into the respective gonads, say testes in case of males and ovaries in case of females. That is what we call migration and proliferation and migration to gonads. After they proliferate and migrate, they become differentiation, what we call differentiation into germ cells. This is very important. Germ cells I have discussed already. Spermatogonia, oogonia. This spermatogonia ultimately develop into sperm. This oogonia become ova. These are the spermatozoa, that means spermatogonia and oogonia, both are germ cells. But these germ cells are coming from primordial germ cell. That means primordial germ cells which originated outside the gonads during the early embryonic development, they proliferate and migrate into the respective gonads and differentiate into spermatogonia if it is male and oogonia it is female in case of females. Okay, then this spermatogonia, that is what we call mother cell, that is mother sperm cell, whereas oogonia is mother ova or egg cells. Later onwards gradually develops into respective sex cells or gametes, say sperm and egg, what we call gametogenesis. This is the whole process, that means the outlook of spermatogenesis. And most important point is the timing of gametogenesis, that means whether these all these processes are same, one and same in both male and female? No, not exactly. In case of males, the spermatogenesis starts from puberty and continue until, that means even for even beyond a particular age, that means old age also. Whereas in case of females, the oogenesis starts during the early fetal development. Not just like males starting from puberty, but they start from fetal stage itself. That means during early fetal development, the oogenesis will begin. But the thing is, the primary oocytes, the resultant product of oogenesis, the primary oocytes, several oocytes will be formed. That means primary oocytes will be formed, but most of them will gradually degenerate. And around 2 million primary oocytes will be there at the time of birth. Around 2 million primary oocytes. In case of females, around 2 million primary oocytes. In case of human beings, okay, particularly for better understanding, 2 million primary oocytes, at the time of birth. At the time of birth, the rest of the oocytes will be degenerated, and it does not mean all the oocytes will at a time mature. We knew that there is a menstrual cycle, and each oocyte undergo respective menstrual cycle. That means one menstrual cycle means one oocyte maturation. So if we count like that, around 500 oocytes maturation. Around 500 oocytes maturation in a female lifetime. Though there are 2 millions of primary oocytes at the time of birth, but they have been arrested, what we call meiotic arrest, at prophase 1 of meiosis 1. Primary oocytes, they arrested, what we called at prophase 1 of meiosis 1, what we call meiotic arrest. Meiotic arrest. During prophase 1 of meiosis 1, they got arrested, what we call meiotic arrest. The primary oocytes, which there are 2 million primary oocytes at the time of birth, they are all under meiotic arrest till up till puberty. Up till puberty. When a female attains puberty, and through each menstrual cycle, one primary oocyte will mature. So like that, around 500 oocytes you can expect. The rest, they may not, because there is a menopause at a particular point of time. But in case of males, it is not like that. There is a restriction with respect to maturity. When a male attains puberty, till he becomes old, he at a particular age of during his old age, he produces innumerable, it's like countless sperm for convenience. It is just like 1.5 million spermatozoa on average per minute he will produce. A healthy human individual, 1.5 million sperms, 1.5 million sperm per minute. Such is the difference between the two. That means 1.5 million sperms per minute from puberty up to a particular age. Whereas in case of females, just one egg maturation per one menstrual cycle. On the whole, 500 oocytes in an average healthy human, female human being. That's the difference. Though the fundamental principles operating in both is almost same, but the timing is different. What we call timing of gametogenesis, that is spermatogenesis, is different with that of the oogenesis. Say spermatogenesis from puberty to up to a particular age in old age, but oogenesis started from early fetal development, so they will start, but they are in a meiotic arrest, and again they will regenerate. That means they restart at puberty, and just one egg, that means one primary oocyte per menstrual cycle. Likewise 500, but in case of males is 1.5 million sperms. That is the difference between the two. Okay, it's about the basics. Let me discuss about spermatogenesis one by one. We have discussed spermatogenesis. It is a sequential production. That means sequential changes that ultimately leads to sperm. Sequential changes that ultimately to sperm. We have already discussed that spermatogonia first become primary spermatocyte likewise. Okay. It again have 3 steps, spermatogenesis. Again have 3 steps. One. Spermatocytogenesis. Spermatocytogenesis. Number 2. Spermatidogenesis. Spermatidogenesis. Number 3. Spermiogenesis. Spermatocytogenesis, spermatidogenesis, spermiogenesis. These are the 3 steps of spermatogenesis. I told you already. Sequential changes that ultimately leads to sperm or spermatozoa. That means one after the other in a sequential change. Say here that is in a simplification. Then we will discuss one by one. First, spermatogonia. Spermatogonia become primary spermatocyte. It becomes secondary spermatocyte, then spermatid, then sperm. This is a classical representation, Spermatocytogenesis means it is synthesis of secondary spermatocytes, simply called spermatocytes from spermatogonia. Spermatogonia to spermatocyte. The word it implies spermatocytocytes. Primary spermatocytes, secondary spermatocytes from spermatogonia. The mother sperm cell ultimately become secondary sperm cell. That is secondary spermatocyte is called spermatocytogenesis. Whereas spermatidogenesis, spermatid, the word it implied. Spermatid genesis means synthesis of spermatid from secondary spermatocyte is spermatocytogenesis. Then spermiogenesis, ultimately differentiation of spermatid into sperm. I told you already. A sequential changes that ultimately leads to production of sperm is spermatogenesis. Spermatocytogenesis, spermatidogenesis, and spermiogenesis. It is not that simple. Just like spermatogonia to primary spermatocyte, second spermatocyte, spermatid, then sperm. It is not that simple. It is a bit complicated and laborious process. For example, we will discuss one by one. Spermatocytogenesis, as we have discussed, spermatid becomes first spermatocyte. That means primary spermatocyte. How? Here, the spermatogonia undergoes mitosis to produce primary spermatocyte, and this primary spermatocyte undergo meiosis 1 to become secondary spermatocyte. Up till this stage it is called spermatocytogenesis. Then the secondary spermatocyte undergo meiosis 2 to form spermatids, what we call spermatidogenesis, and this spermatid differentiates. That means structural differentiation, more specifically, or what we call anatomical differentiation of spermatids into sperm or spermatozoa, what we call spermiogenesis. So ultimately sperm. Here the thing is spermatogonia, very important. Spermatogonia again of 2 kinds. Spermotype A and type B. How they form primary spermatocyte? That is what I am explaining. Type A and type B. Type A again of 2 kinds, AD and AP. That means depending on the color of the nucleus. If the color of the nucleus is dark, this is called AD. If it is pale, this is called AP. Okay, depending on the color of the nucleus. Okay, and AD, this is called self-renewing nucleus. Self-renewing type. Okay, self-renewing type. The name it implied, self-renewing. Either it may produce AD or AP. AD or AP. Okay. Whereas AP is called intermediate spermatogonial cells. AP is called intermediate spermatogonial cells. And this AP become, that means this AP cell divides to become B spermatogonial cells, and this type B spermatogonial cells are called committed cells. committed spermatogonial cells. Committed spermatogonial cells. And this committed, the word in bracket commit, what exactly it means? This type B spermatogonal cells become primary spermatocytes. They become primary spermatocytes. Spermatocytes. You know, that means this one. I told you already. Spermatogonia to primary spermatocyte is that simple. Again, it is having a laborious and complicated process. Type A and type B. Again, depending on color of nickel, type A, let me AP, let me AD, A dark, AP, pale. Dark means it is self-renewing, either it becomes AD or AP. AP is an intermediate spermatogonal cell. They become B spermatogonal cells, and these B spermatogonal cells are otherwise called committed spermatogonal cells. They become, that means they divide to become ultimately primary spermatocytes. That means B spermatogonal cells are the committed, they become primary spermatocyte. That is the mechanism involving in spermatogonia to, that's the first step, spermatogonia to primary spermatocytes. And these primary spermatocytes, which are the products of mitosis, you know, mitosis, undergo meiosis 1 to become second spermatocyte up to this called spermatocytogenesis, and they become meiosis 2 to form spermatids. Then either haploid, secondary spermatocytes are haploid, primary spermatocytes are diploid, spermatogonia are diploid. Haploids are secondary spermatocytes, spermatid and sperm. That means the spermatids are the resultant product of meiosis 2. Spermatid, they undergo differentiation. Just like primordial germ cell differentiate to become, you know, spermatogonia, the spermatids differentiate to become sperm. That's it. So ultimately, these were about the sequential steps of spermatocytogenesis. That means spermatogenesis. Okay. Then we will take a diagrammatic outlook for better understanding. You know. Say this is a spermatogonia, a diploid spermatogonia. It becomes primary spermatocyte. The mechanism we have discussed, primary spermatocyte. This primary spermatocyte undergo meiosis 1 to form two haploid secondary spermatocytes. And they undergo meiosis 2 to form spermatids. Then these spermatids, these haploid, you know, these are just 2n and n haploid. They ultimately differentiates to form sperm cells. You know, spermatozoa. Okay, this is a simple representation of spermatogenesis. Diploid spermatogonia become, that means undergo mitosis to form primary spermatocytes. They undergo meiosis 1 to form secondary spermatocyte. They undergo meiosis 2 to form spermatids. The spermatids, they differentiate to form spermatozoa. So the first, here again the first step, spermatogenesis we already discussed. Spermatogenesis, just directly the secondary spermatocyte, they become spermatids. Spermiogenesis, here spermatids become, that means differentiate to become spermatozoa. The third step is very important. You know, we already discussed differentiation. You know, differentiation. What exactly differentiation? The third step is very important. Third step, what we call spermiogenesis. What exactly means the differentiation? For better understanding, we'll draw a sperm diagram for convenience. You know, this is a condensed nucleus. This is the acrosome. This is the acrosome. This is the middle piece. This is a long tail. You know, proximal centriole. This is the distal centriole. Distal centriole. Then mitochondria are spiral shaped structures. Spiral mitochondria. Nebenken, what we call Nebenken. Then tail, flagellum. This is head. This is neck. This is midpiece. This is tail. And this is acrosome. This is nucleus. It's a basic spermatozoan structure. Okay. 4 parts is the structure of spermatozoa: head, neck, midpiece, and tail. Head consists of acrosome. Actually, what is that? What exactly acrosome? Golgi complex. The nucleus. Here, the nucleus actually condensed, transcriptionally inactive. This is very important. Protamines, they replace the basic proteins in DNA, and so that they become transcriptionally inactive in condensed stage with an anterior, like oval shaped structure, where there is posterior there is a depression, and depression contains proximal centriole, and this proximal centriole will lead to spindle formation. Spindle formation useful during first zygotic division. Whereas distal centriole leads to axial filaments. Axial filaments. Distal centriole leads to axial filaments. Axial filaments are microtubules in 9+2 element, whereas in centriole just 9+0 element. And that means axial filaments can ultimately the flagellum, tail. Whereas the significance is, the mitochondria in spiral shaped structure. The Nebenken, what we call. There is spiral in mitochondria. There are additional structures like galea are also present in, just I am not going into the details, and manchet are also present. That means at the anterior acrosome there will be galea, like galea. And the posterior, that means posterior part up to midpiece, manchet. So this is basic spermatozoan structure, but it is matured spermatozoa. It is not that simple, Differentiate, just I have used the word differentiation. How the spermatid differentiate to become spermatozoa? Here the thing is, we have different cell organs like acrosome, Golgi complex. They become acrosome. Centrioles, the proximal centriole, this leads to spindle formation, and after fertilization, the first zygotic cleavage is based on the spindle formation based on proximal centriole. The distal centriole becomes, leads to axial filaments, that is to tail formation. The mitochondria in the form of spiral shaped structure that form middle piece, and the neck is a depression like structure that consists of distal centrioles, and the condensed nucleus and galea like structure. So these are all differentiations. What we call spermatid actually is up till spermatid. That means from spermatogonium to spermatid there are incomplete divisions because they are all held together by cytoplasmic bridges like this. They are held together by cytoplasmic bridges. Here, the mature spermatozoa should detach from it and get into the lumen of the seminiferous tubule. This is called spermiation. Spermiation. Liberation of matured spermatozoa into the lumen of the seminiferous tubule is called spermiation. From where? Sertoli cells. This is the most important thing we want to discuss when during this point. That means that the differentiation spermatogenesis, Sertoli cells. Sertoli cells are simply nourishing cells. Nourishing cells. The most important is androgen binding protein, ABP, androgen binding protein. They synthesize androgen binding protein coming under the influence of follicle stimulating hormone. We knew that pituitary, anterior pituitary will release follicle stimulating hormone FSH, and they will stimulate the Sertoli cells to release androgen binding protein, and this androgen binding protein will concentrate the testosterone so that it leads to the maturation of spermatozoa. There should be an environment. The proper facilitation of environment should be there, and it will be provided by sperms on the Sertoli cells. Say androgen binding protein, it will concentrate testosterone for the maturation, that means for the development of spermatozoa. It also maintains blood testis barrier, that means what we call facilitation of environment for the spermatozoa formation. That means it's like nourishment structures, and most importantly, it will provide ABP and also synthesize, it will secrete testicular supporting fluid. When the sperm detached from the Sertoli cells into the lumen of seminiferous tubules, they are non-motile. Though they are mature but non-motile, they doesn't have the capacity to fertilize. They have to reach the epididymis, where they become a motile capable of fertilization. For that, they require testicular supporting fluid synthesized by the Sertoli cells, which will reach the epididymis. There they become motile. This is very important point. And these Sertoli cells, this like supporting cells in every sphere. They provide environment, they provide hormonal environment, they provide testicular supporting fluid so as to reach the epididymis. In epididymis, they will attain the fertility. That is a capability of fertilization. This is very important. This is metal plus maturity. This is very important. Okay. So these are about the Sertoli cells. That means the spermatogenesis is under the influence of hormones, local temperature, and temperature and the local androgen binding protein, Sertoli cells. And most important, sometimes there should be a regulation in FSH, follicle stimulating hormone. Otherwise, leads to excess production of sperms. So to cut the same, there will be inhibin released by the Sertoli cells. This inhibin will control the FSH. So it's like negative effect. Though the FSH stimulates the Sertoli cells to release ABP, if there is excess count of sperm, this Sertoli cells release inhibin that control the FSH to regulation of spermatozoa. So it's about what we call different factors that regulates the spermatogenesis. Okay, thanks for watching.